All right, so it's my honor to present on behalf of the Carcinogenomics Working Group, as well as my co-chair Chris Corton today on our updates on progress this year. Next slide. So we have a really nice list of contributors across industry, academia, and government. So big thank you to everyone and all the hard work that was done this year that we'll be talking about. Couldn't do it without everyone. All right, so this work stems out of a vision really around supporting the new ICH guidelines and what can we do to build a better way to evidence early on in short-term animal studies before going into 2-year carso studies of potential tumorigenic risk of unknown compounds. And so two areas we're focusing on is very early on and short-term, say, under 1 week studies, can we see induction of mechanisms that we know will lead to tumors over time? And then second, in slightly longer, say month-long studies, can we see early evidence of clonal expansion um, that over time would lead to tumors, but can't detect yet histologically, but can we use some sensitive techniques to detect them sooner? So this is really the strategy that was laid out in this paper by Chris et al. In 2022 from the HESI working group. And so we're presenting on the progress we've made on these fronts, both in this working group as well as the error-corrected sequencing working group, which is up next. So Connie, next slide. So then more specifically on the early markers, the ones we hope to see within just a few days of dosing, we've been focused on transcriptional signals since many of those molecular mechanisms are transcription factor based. We can look at the downstream genes as potential biomarkers. And that's the focus of the Carcinogenomics working group. And then the error-corrected sequencing working group is focused more downstream. Again, we'll be presenting next, and that's focused on detection of clonal expansion at a slightly longer time frame within months. So just specifically then the objective of, Connie, just to go back, the objective of the CGS working group is really to drive industry, regulatory, and academic understanding and acceptance around this weight of evidence approach using new genomic biomarkers that could be applied in vivo short-term rat studies to inform on risk after a 2-year carso study. Now, next slide. All right, so specifically then, where we're focused in the carcinogenomics group, there's multiple AOPs that we believe lead to rodent tumors that are non-tumorigenic. So these include things like nuclear receptors, AHR, CAR, PPAR, persistent cytotoxicity, and then of course direct DNA damage. And we know from prior work, I think there's a unknown automation here that's missing. Connie, if you click once again. There we go, the references. We know from prior work by Chris's group, by Merck, et cetera, that a number of these transcription factors you can develop gene signatures for, and these show early promise of being predictive of long-term outcomes in the rat. And so really focused on developing the biomarkers of the activity of those receptors. We're focused in liver since that's where we have the most knowledge, but also where we have large external data sets that we can use to develop these gene sets. Next slide. So this is how the working group is organized. We obviously have a strategy and outreach team, but then a lot of the upfront work and really the focus for the first couple years of the working group was just to get the data to a point where we could use it to develop signatures. So we had a data compilation team, which pulled in these large external genomics data sets on livers treated with various compounds. And then we had a mechanisms and compound classification team, which took the compounds for which we had data and annotated them both by what MIEs they're known to interact with, right? Are they AHR activators, et cetera? And then also bringing in the 2-year carso study data for each of those compounds and relevant doses. So are they associated with liver tumors, other tumors, and at what doses? So like I said, that was really the first couple years, and then over it was about the last year and a half, the fun really begins, where the gene signature team was able to really get off the ground, leveraging all that data to start developing the markers for each of the MIEs. So I'll just walk through some of the progress this year on those various teams. And so, yeah, the next slide. As far as outreach, um, you see a nice picture there of our esteemed Connie Mitchell presenting at SOT. So she provided an oral presentation on the work at SOT. We also had a CSS webinar in February that outlined the work. And we've also been drafting up a manuscript on this gene signature identification. While that work's still in progress, we have the framework of a manuscript, so we hope to be able to move that quickly once we reach final consensus on the gene sets. Next slide. The data compilation team is really complete at this point. You remember from previous presentations, all the work that team did. We took 6 rat liver, AFI, and RNA-seq data sets from an external domain, which covers 370 unique compounds treated to rat liver. All the raw data was brought in-house, processed, normalized, QC'd, and then hosted both on SEBS and then in a SQL database that we use to distribute to the team and the signature identification team for their analytical work. Also, prior years, the compound classification team had done all the MIE annotation. So which of those compounds we know induce any of our key molecular initiating events. So that's all been annotated nicely. And then in 2024, that team has been completing the data on the 2-year Carso studies. So we've brought in most of that data through the GOLD database, ToxRefDB, or LASA. And so much of that work is now complete. And the next step is really aligning the 2-year Carso study doses with our genomics data and which treatment groups align with which outcomes in the 2-year Carso studies. So hopefully those efforts will be finishing up shortly. Next slide. I'm just flashing up here where we pulled the data from. Again, it's a total of 370 compounds, and it comes from mostly drug matrix, TG Gates, Bayer, some of the validation work that SEQC did using drug matrix, and then a Merck study, which is unique, and then it's RNA-seq, and so it's a nice way of validating the signals that are seen across platforms. Next slide. Okay, so presented this slide before, but just to again highlight the strategy we're taking for biomarker ID. So we're taking focusing on training signatures using all the AFI data, because that's where we have the most compounds for each individual MIE. We're taking compounds known to induce a specific mechanism, looking for signatures or genes that are sensitive to those specific mechanisms. So that's the first column there in A. But then also ensuring those signatures are unique to that mechanism. So often when you do these approaches, you'll get some genes that come up in multiple MIEs, and so being able to weed those out by looking at the bulk of the data and making sure that we're looking at genes that only come up with the compounds that are known to induce that specific mechanism. And then the third column is a really nice study that Chris Corton spearheaded through this working group, where we're taking rat knockouts of each of the, I believe, 6 key MIEs, treating both wild type and knockout animals with paradigm compounds, and then performing transcriptional analysis, again, to confirm that the biomarkers are specific for each of the MIEs we're looking at. And we're also hoping to do some ChIP-seq work there to confirm that the downstream genes, right, focus those on the direct targets of the transcription factors of interest. So I'm going to talk about first the knockout study where Udayan lab has made really nice progress this year, and we're almost complete with dosing all of these studies. So that's shown here. So we have wild type in knockout and wild type controls for each of these MIAs, P53, CAR, PXR, PPAR alpha, AHR, and ESR1. Everything is complete except being dosed now is the AHR study, as well as we're still missing 2 females from ESR1. Those have been hard to breed out, so hopefully we have those wrapped up soon. But really nice progress, ton of work by Katie et al. In Udayan's lab, so big shout out to them. Just to note that based on feedback from this group, a number of different tissues were collected, as you can see listed there. Pieces of the liver have been sent to Merck for transcriptional analysis, but those samples are in freezers and open to discussion on interesting ideas of how to leverage those other tissues as well. Okay, so here's I want to quickly flash some of the late-breaking data we have here. So far we've completed RNA-seq for PPAR, P53, and ER. PXR and CAR are on the sequencers as we speak, so we'll have that data soon. But so far it's been textbook data coming out of these models. It's really exciting to see. I'm just showing here published gene sets from Alexi's paper. We'll obviously be mapping our various gene sets onto this data. But we can see on the top of each heat maps are the wild type animals treated with respective agonist of each transcription factor. You can see really strong induction of known signature genes by those agonists. And then when you look in the knockouts, those effects are pretty much completely gone. So real exciting. Really, I think confirms the proof of concept of what we're doing here, and the ability to identify genes that are, hopefully nice and proximal to these transcription factors. Next slide. Okay, so on the gene signature development front, just to sort of level set of where we are, there's multiple steps we need to take. The first is to define what are the genes that are responsive to each of the transcription factors of interest. And that's really where we are still. Once we define those gene sets, we can then start defining point of departure. What's the variance at baseline? When do we see signal that we trust is indeed an activation of that transcription factor? But we also know that low levels of induction of these factors are not an issue. So the next step will just be able to just confirm that strong activation of the receptors are indeed predictive of outcome at 2 years. And if so, then what level of induction, is it, there's a specific degree of magnitude of the signature? Is it sustained response? Those sorts of analyses will need to be performed as well. The good news is the signature identification is the hard part, and once that's completed, I expect the rest of the steps to move rather quickly. So we're almost there. All right, so progress on the gene signature identification work. We presented last year when we met that we parsed out the data to various different groups from the working group, including NIH, EHS, Takeda, Merck, Pfizer, GSK, and really allowed each of these groups to take their own approach to the data. We wanted to see, we didn't want to bias up front this analysis. We wanted to see if folks took this data on their own, what they would come up with and how those data would overlap. So last year we had just received preliminary results back from these various groups, and we're really excited to see that indeed we were seeing overlap in the responses. And then this year we've been working to really, based on those results, align our approaches across the groups, making sure we're using consistent treatment groups, et cetera, to shore up those signatures. So I believe, Connie, on the next slide is our current, yep. So what we're showing here, again, each group went off and developed their own signatures for each of these MIEs. And then we compiled all that data and just asked which genes were being detected by multiple groups. And so what I'm showing here are genes that were in signature sets from at least 3 of the 5 groups. And you can see we have a number of genes for each of the MIEs where we're seeing consistent findings. And if you look closely, very interestingly, we're seeing a lot of our known friends from each of these MIEs. So CYP1A1, 1A2 for AHR, the ACODs, ACOXs for PPAR, etc. So these signatures, not only are we seeing nice overlap between the groups, they're making biological sense. So really exciting to see. Next slide. So, fully transparent, the hard part now is really aligning on what are the final sets. You you have different analysts in the room. There's always different ways of approaching the data, and so coming to consensus across the working group on these final sets is not an easy task, and it's really where we've spent much of 2024. So just some details there. What treatment groups do we use? Some of the MIEs come up later, some come up early. So, do we use specific time points for a specific MIE? Making sure we're comparing sensitivity and specificity outcomes, not only in the AFI data, but then mapping that onto the RNA-seq data to confirm those genes are performing on RNA-seq and that they remain specific. Another thing is just setting thresholds for signature identification. When you sort of give out data and ask folks to come back with signatures, some groups come back with very short lists, some groups come back with very long lists. And so what's the optimal size for each MIE? That's something we're working through. Obviously, adding on the knockout data now that that data is rolling out, that'll add into our specificity analysis and sensitivity analysis, of course. And then with all the above, hopefully within the next several months, if I'm optimistic, we can align on consensus approach, final gene sets, and then finish up the manuscript that I mentioned is being drafted. Slide. All right, so last slide, just finishing up as a reminder of that's part one, identifying the gene sets. Part two will be identifying the levels of activation that are associated with tumor risk. And so, once we have the gene sets in place, this should be relatively straightforward, but acknowledging that that'll be the next place we need to go and aligning with all the 2-year outcome data and relevant dose data that's been compiled by the compound annotation group will really be the next step. So Connie, do you want to take the polls from here?
Yeah, we had some discussions with Keith and with Chris on, how we're going. I think 2024, we had a lot of really good progress, but it was a little bit cyclical. So we just kind of wanted some feedback from the team. So similar for before, if you can't access the chat, I'm sorry about that for folks, or you can't access the polls rather. There's a few polls. We just wanted to hear, what are you excited about? What do you like that this team has done? What for you is the most meaningful that has been done so far? So hopefully people can start to see that and answer. Let me just see. So at least one person responded so far. So those are coming in slowly. As those go, I think we have a couple. We just want some feedback. The next thing is for 2025. This project has been going for a few years. It's been really good work, but wanted some feedback on where we should focus our efforts. So I'm going to launch that one too. So folks still answer the first one, but going to continue to launch these. So, and if you have other ideas, feel free to put it in the chat, or if you can't answer, but one thing we're planning and trying to do is publish the results, but if we should really focus on that, I think we could put more effort towards it, adding or refining the thresholds. One thing, once we have the biomarkers, how do we actually, maybe small v going to big V validating them? So like looking for samples that already exist. This is really in the context of, short rat studies. So to actually run a lot of those would be expensive for ourselves, but possible if we really thought that was, the main focus. But I think if we could leverage existing samples similar to what we're doing in error corrected, that would be great. Yeah, I see some comments from Carol and some folks that can't maybe access. And then the last one, again, a lot of these collaborative projects, they take time, right? And sometimes it's important to build consensus, but that's not necessarily quick. So we wanted to hear if there were ideas that could help us move a little faster. They're open to different ways. Sometimes in projects we've had a postdoc give a little bit of their time. That might be something that we're utilizing with Carol's lab, two different postdocs actually that have been really great to help move things forward. Is there ways we could, use a statistics service to get things done, kind of open and, what we can do and what's actually practical is based on the funds we have, which isn't unlimited. But if there's ideas to help move things forward faster, I think we're open to it. I don't know, Keith, if you'd like to add to any of those.
Yeah, no, on that last point, I meant to mention that during the presentation that, we realized that having data in the hands of various different groups and trying to reach consensus that way is difficult. So a really good idea, I don't know, Connie, if it's your idea or whose, but was to recruit Srijit Seal to help. So who's a postdoc at the Broad who's helping us, have an external set of eyes on the data, bring the various approaches from the different teams together in order to sort of analytically help us reach consensus versus having our data in various pieces and not having a concerted analysis. So I think that'll be really helpful to have one person who can bring everything together and help us, look at the consensus findings and reach a final set.
Yeah, well said. We're getting some ideas from Steve to try these in human cells. I think that's been something we've wanted to look at least how do they perform, knowing there will be some species differences, but that's always been a next step. It's just trying to get through this part first. But it's something once we have the biomarkers, it's worth a look, I think. Yeah. So I think those are the polls. Feel free, folks, to continue to put things in the chat or answer those. We can have a few minutes for questions as well. Really, thank you, Keith, for walking through all that.
Thank you.
So,
thanks to the group. It's great progress this year.
So folks can either raise hand or put in the chat if they have any questions. I don't know if there's anything Chris wanted to add as well. Oh, I see Udayan popped up. Go ahead, Udayan.
Udayan's the star of the show.
No, we had great fun doing this work, and thank you for all your comments. But please take the tissues. I mean, there's a lot of stuff there. These were hard studies to do, and there is wealth of data in there. I always sit there wonder about extra hepatic tissues, and I'm a self-confessed hepatomaniac. So anything else is kind of there to support the liver in my head. But I'm sure people are interested in other tissues like lungs. We have lungs from all of these things, testes and other organs. And I'm sure these NIP2 receptors drive biology there. So there's so much to learn from this. So that's one thing. I just want to put that plug out there. But my question to, I guess, the group is, the ultimate goal, I guess, is to somehow convince the agencies to sort of adopt this. And I'm just wondering what is the game plan in the sense, where do you go? Maybe a broad picture is fine, no details, but how do we reach that goal from where we are now? It's what, Connie, if you had any comments on that, sort of.
Keith, did you want to start? I can start.
Yeah, or Chris, anybody, yeah.
Sorry, as far as reaching consensus?
I think,
no, I mean, convincing the federal agencies to adapt these to actually use them. What are the steps we can take to kind of get there?
It's a good question. It's something we had discussed in formation of this working group is, how far we would take these. And I think we realized, particularly with examples from the past, how long of a road that is. So we've really been focused on establishing the markers, publishing them, less so on next steps. But I'm open to input from folks on how far they think that, HESI could take this.
Yeah, and I think we might have mentioned earlier, but there's going to be the development of them, but are they actually working? And some of that might be with known samples that people have in their freezer or known data sets that we didn't tap into yet. It might be that people start to run these. So I know for, say, the T-pod, people are starting to run, say, 5-day studies, in rodents. So that might be an opportunity if they're already doing the sequencing to look for the biomarkers as well. So it might pair with that or some of the other short-term studies that are required. So I think there's that confidence building step, and yeah, how much we take on as an effort we'll need to discuss and be, but it's kind of like putting the tools out there, but also how do they work? What refinements might be needed? How do we get these taken up as maybe panels that are available? So I think there'll be like kind of needs of the community to try and see how these are working. I'm open to other ideas and suggestions.
I think the new ICH process will also help as these roll out. Seeing really nice pass forward there with companies submitting for the not needing to do your 2-year carcinogenomics studies, and obviously this data would help companies write those white papers, and so that might be also one way of folks getting confidence and familiarity with these markers over time if companies start using them in that weight of evidence approach.
I will also just say, I think it's right to think how companies can use this, but it's not the only people who can. There might be some folks that, maybe academics that want to know about mechanism. These could be good tools for things like the National Toxicology Program or DTT. It might be a way if they're doing these shorter term studies, or like EPA as well, doing shorter term studies to try and bring in that mechanistic info without having to do the longer term, or maybe as a way to help prioritize what you would do maybe a 2-year study with. And Patricia is about to speak too about, with error corrected, how these tools could be used together. So I appreciate the comment, Shudai, and I think it's important to say, this isn't just an academic exercise, but how people use the tools might be a little bit based on their questions.
Right. No, thank you so much for that.
Yeah. So I think for time, that might be it. Anything else you'd like to add, Keith, before we wrap up, or Chris?
No, happy to field questions, follow-up questions if anyone has them later.
Great. Let's give a big hand to Keith.